There we
see
go.
me now?
Yes, we can.
All right. Yeah. So you might be interested in communication between different cell types. And this is a true story. I talked to someone where that was their major question. They were interested in the communication between glia and neurons, and they only had data from glia. They didn't collect any data on neurons. It's like, well, sorry, you can't answer that question because you didn't collect the right data. Or you might be interested in sort of transcription factors and gene regulation. We're not going to really talk about that too much because that's best answered doing single cell ATAC-seq and or multiome, so single cell ATAC and RNA together. And we're focusing on RNA-seq. But again, that's something, right, you need to figure out if that's what your question is, collect your multiome data instead of just RNA-seq data. So if we go back to our clustering workflow, right, we've done our normalization of normalized expression. We've talked about feature selection. We've talked about dimensionality reduction. So now we're going to look at cell-cell distances, making a graph, and then clustering trajectory inference and differential expression. So this is really where we're answering our biological question. That's cool. So the reason we're having to do all of this is because to do our trajectory inference and our clustering, it's much easier to do that for a big data set if your data set looks like a graph. So the best clustering methods for very big data sets were invented when social media started being a big thing, and we had giant data sets of friend groups and interactions between people. That was a graph, and people developed lots of clustering methods for big data based on a graph. And now it's just much easier to turn our matrix of data into a graph and use one of those methods than try to develop a new clustering method that will work well for our matrix of high dimensional data. So what do I mean when I'm talking about a graph? So a graph is something that looks like this. It's made up of two things. One is vertexes, also known as nodes. In our case, those are representing individual cells. And edges between them, which represent some sort of relationship between those two things. So for our case, what an edge means isn't super clear. We're going to define our edges as cells are similar to each other or not similar to each other. So the way we create our edges is using k-nearest neighbors. So what k-nearest neighbors means is we take each cell. So you can imagine these is our data in PCA space. And we're going to connect each cell to its k nearest neighbors. So in this case, if we use k equals 3, we could take that cell and connect it to its 3 nearest neighbors. That cell connected to its 3 nearest neighbors. Repeat for all of the cells, and we'll get something that looks like this. Okay. Pretty straightforward. However, you'll notice we have some problems. So we have these cells in yellow that look like outliers, yet they're connected to multiple cells in our data set. So that's not ideal. We'd really like these outliers to stay separate, stay away from our other cells because they're outliers. We also have this group of 3 cells that are connected all to this other group. Right? That's because our k is 3, so each of these had to find a neighbor outside of its cluster. So if we're looking for small clusters, we need to be careful about what we're setting our k to, so we're not merging a small cluster with other clusters because our k is too big. The other solution to both of these problems is what we actually do. So I just lied to you. We don't actually do k nearest neighbors. What we do is shared nearest neighbors. So what shared nearest neighbors does is it takes our k nearest neighbor graph. So it does that first. We do our k nearest neighbor graph using our k as normal. And then it goes through each node and asks, between you and one of your neighbors, how many of your neighbors do you share in common? So hopefully Ottawa can see my cursor. So if we consider this cell here and this cell here, right? They have two neighbors in common with each other, right? These two cells are neighbors of both this cell and that cell. So the edge between those two cells would now become the number 2 because they share two neighbors with each other. And we just do that for every pair of cells, count out how many neighbors they share with each of their neighbors, and weight the edge based on that. And you can see for things like outliers, right? This guy out here, it's connected to these 3 cells, and they don't share any neighbors with each other because they're all in disparate parts of our graph. So these edges will all get erased. They'll all get set to a weight of 0, and our outlier will correctly be identified as an outlier and not connected to the rest of our graph. Similarly, this rare group over here, the small cluster, they all share that cell as a neighbor, but that cell doesn't share any neighbors in common with them. So again, those edges will get erased, and this small cluster will be correctly separated from the other cluster. Right, so we're going to use this approach to have sort of nicer edges between our cells and clean up our graph a little bit from doing k-nearest neighbors. So once we've got this graph, how are we going to define clusters on it? So whenever we're doing clustering, we have to define what do we mean by a cluster, right? That's just a word that doesn't actually mean anything until we define what it is. So for a graph, we define clusters using this score called modularity. It's not super important. This is the equation. I'm not going to go through the details of exactly how this works, but essentially what this is doing is it's looking at A, which is the observed number of edges between 2 cells, and subtracting from it the expected number of edges between those 2 cells, and then adding that up for all of the cells that are within the same cluster. So this is defining our clusters as a group of points that have a high density of edges between them and few edges between different clusters. Right? So that's just how we're mathematically defining what a cluster is. So for instance, if we had a graph like this, and the clusters were our green and purple here, we would get a pretty high modularity. We get a modularity of 0.4, which is pretty good. Whereas if we clustered it like this, instead, our modularity will be negative 0.1, which is a bad score. So the bottom would be bad clusters, the top would be good clusters by this score. Okay. So hopefully that seems intuitive that that is doing what we want it to do. So all we're going to do now is take our score that defines a cluster and try and optimize that score across our cell-cell graph. And once we've optimized that score across our cell-cell graph, that will be the clusters we get at the end. So the way we do that optimization is using what's known as the Louvain algorithm. It's now often also the Leiden algorithm, which is not quite the same as the Louvain algorithm, but it's very, very similar. So we're just going to do the Louvain algorithm for now. So essentially how this works is you have your graph, and you start off by labeling every cell as a separate cluster. Each one is unique. And then we're going to iteratively go through and randomly pick a node and ask, given the neighbors of this node, if I change the color of my node, so the cluster ID of this node, to the same as each of your neighbors, what would give me the best modularity score? And when it finds which one gives it the best modularity score, it changes the label. So that, in this case, that cell would become blue. And it just keeps doing this, picking random nodes over and over again. So this one turns pink, this one turns blue, that one turns blue, that one turns pink, etc. Until eventually it gets to the point where it can't change the labels anymore. It's already reached the optimum. And when it does, it stops and says, here are your clusters. Okay? So that's how it's working. Again, I did just briefly lie to you, or because I glossed over one thing, which is the resolution parameter. So you'll see when we run Surat, or if you're doing any other implementation of the Louvain clustering algorithm, you'll have this parameter called resolution. And what that is this little symbol in there. So it's affecting the relative weight of the number of edges we expect by chance. So if you have a resolution parameter of 1, you want your clusters to have more edges than you expect by chance. If it's less than 1, you're allowing some degree of less than random chance edges to still constitute a good cluster. If it's higher than 1, you're being even more strict. We also have the parameter of the k we use to create our shared nearest neighbor network. So for a resolution parameter, if it's high, we're putting a lot of weight that we need really a lot of edges to be called a good cluster. So we're going to get many small clusters if we do that. If we have a low resolution parameter, then we're being more permissive and we'll get a few number of larger clusters. For our k parameter, if we have a large k, each cell is connected to many other cells, so we'll get a small number of large clusters. If we have a low k, each cell is connected to a few other cells, and we'll tend to have smaller clusters and more of them. There is no optimal setting for what these should be. Various people have tried to optimize it across a bunch of data sets, and essentially applying it to standard 10x data sets. It seems like a resolution parameter of 0.8 and a k of 20 seem to work good. In the general case across many standard 10x data sets. But if you're working with a weird organism or you're looking for a particularly rare cell type, these defaults might not work for you. You might need to adjust them. Yeah. So k high is few large clusters, low is many small clusters. Okay. So that brings me to discussion of rare cell types are hard to find. So why might they be hard to find? Where in this workflow might they get lost?
They don't really have neighbors. They don't really have enough neighbors.
Yep, so Edmonton suggests they won't have enough neighbors, so that will, if our k is too high, that will mean they'll get merged to another cluster, so that we won't see them anymore. Ottawa suggests dimensionality reduction. Yeah, so if we're picking the top 20 principal components, a rare cell type might not be captured in those top 20. It might be down in principal component like 50, right? So you might lose it there. You can also lose it in feature selection. So the marker genes for a rare cell type might not have very high variability because there's so few cells expressing them. So they might get filtered out there. They can even be filtered out if you apply a threshold of like a gene has to be expressed in 1% of all of your cells. Well, a cell type that's less than 1% of your cells, all the genes that are unique to that cell type are going to be filtered out. So there's a few places we can lose our rare cell types. So again, if you know your data set and you're trying to find these rare cell types, just be aware and go back and check all of these steps and maybe adjust the parameters to help you find those. All right, so now that we talked about our clustering algorithm, what are the limitations of our clustering? So a clustering algorithm will always give you clusters. That doesn't mean those clusters represent real cell types. Okay? And that's true for all of the algorithms. So pseudotime analysis or trajectory analysis, also, they'll always give you a trajectory. Doesn't mean that trajectory is real. So here's a little example. So here's some data. How many clusters are in this data?
Depends on how we set the modularity, right?
No, there's a true number of clusters. I generated this data based on there being a certain number of clusters. How many do you think I used? So Ottawa has some votes for 5, also for 4. Edmonton seems to like the number 4.
Fine.
5. Vote for 3 as well in Edmonton.
Can we give like a confidence interval, like 4 plus minus 1?
Edmonton wants to go for 4 plus or minus 1. Nia also likes that, wants that to be an option. Ottawa seems to like number 5 clusters. So this is my guesses on how you might split this up into different numbers of clusters. So you can see how you could get 5 there. This is the correct answer. So there was actually 4. But you can see, even for humans, with a very simple 2-dimensional data set, this is a hard question. So when you're looking at your single-cell RNA-seq using an algorithm in super high-dimensional space with your UMAP and t-SNE approximations, this is going to be a very hard question to answer. So how might we try and answer this for our single-cell data set? So there's lots of ways to try and determine what the right clustering is for your data set. And the reason there's so many of them is because none of them universally work or are the best. Right? They're all different ways of measuring it. They all have pros and cons to them. So one of them is to look at the robustness of the clustering to methods or parameters. So you can change your K, you can change your resolution, and see if you still get the same clusters. You could look at marker genes or differentially expressed genes between your clusters. Right? So if your clusters are real and represent different cell types, you should be able to find marker genes that define those 2 cell types that are different between them. You can also use known marker genes. So if you're looking for particular cell types that have known marker genes, you can look to see if those marker genes are different between your clusters. There's a variety of quality statistics. So we're going to talk about the silhouette index, but there's some others that give you a numerical score for how good are your clusters based on some particular statistic. You can also look at how consistent they are across experimental replicates. So you could cluster your disease case and your healthy case separately, or you could cluster 2 replicates of your data set separately and see if you get the same set of clusters, right? The same set of cell types. If you have matching spatial transcriptomics data, you could look to see if your clusters follow different spatial patterns in there to validate them, or you can do some sort of experimental validation of those clusters. However, that's very slow and time-consuming and expensive, so it's usually the last thing you do after you've tried everything else. Note that seeing that they look good on your UMAP is not on this list. That's because UMAP uses all of the same assumptions and often the exact same parameters as your clustering method. So you're almost guaranteed that your clusters will look good on your UMAP, even if they are not true different clusters in terms of cell types. And this is very often for most clustering methods, that they'll come packaged with a visualization that is all but guaranteed to make those clusters look good. Any questions about that? Doesn't look like it. Oh, yeah?
Reference data, like Are there reference data?
Yeah, so that's another one. So Edmonton suggests reference data. So that's another one that sort of goes with number 5 here, is you could compare it to reference data. So whether your clusters represent known cell types from reference data.
No, I didn't know that the reference data exists. Would that only be like for humans or?
It depends. So there's a lot of reference data for human and mouse available from the Human Cell Atlas or on things like the Tabula Muris or the Mouse Cell Atlas, or there's like mouse brain atlases. So there's a lot of atlasing projects that are nearing completion now. So there are reference data sets for most normal tissues in mouse and human. If you're using something other than that, it's unlikely there'll be a reference data set available that you could compare to. So what's the silhouette index? So the silhouette index is the most popular method for calculating your cluster quality. So the way it works is, so yellow and blue here are our 2 different clusters, and the dots are our different cells in our cluster. So for each cell, it calculates the average distance to the other cells in the same cluster as it. It then calculates the average distance to the cells in the next closest cluster to it. So in this case, we've only got 2, so it's going with blue. And then you just take the difference between those and divide it by the maximum of those two values. So if you get a score of 1, you're much closer, you're essentially perfectly close to the cells in your cluster and very different from the cells in the other cluster. If you get a score of -1, you're essentially identical to the cells in the other cluster and not like the cells in your cluster. So this gives you a score for every individual cell for how well it is clustered with the rest of its cluster. And then you can aggregate that at the level of cluster or the level of data set. So you can calculate the average silhouette across each cluster and see how good each cluster is individually, or you can average across the whole data set to see how good is your clustering in general. So here's a little example of what this sort of looks like. So here's a relatively small data set. On the left, we have the cell-specific scores for the silhouette index for each of the clusters. So they're colored by the cluster, and there's a bar for each cell. On the right is the UMAP or PCA plot of the data, right? So that's the visualization of the data and the clusters. So you can see in this case, we have relatively distinct clusters. So we have positive scores for the silhouette index for all of the cells and all of the clusters. And we have good scores, which is conventionally higher than 0.65 silhouette score for many of the cells and many of the clusters. Whereas if we increase the resolution, one of the clusters down here is now cut into 2, split into 2 separate clusters. And you can see on the silhouette scores, the silhouette scores for those 2 clusters now is much lower than for the others. So this would be a hint to you that those 2 clusters are not very distinct from each other. So they're not as good clusters as your other ones are. There also is the problem that in biology, there's not one correct answer to the number of clusters you have in your data, right? You can define cell types at different scales. You could have one set of clusters that represent T cells versus macrophages versus epithelial cells, and a different set of clusters that represents CD8 positive T cells versus CD4 positive T cells, and mucus producing epithelial cells versus basal epithelial cells, right? And those are both perfectly valid and equally good sets of clusters.
Quick question, that how we can set a threshold for this kind of clustering? Because it's kind of a way that I know that it depends, as you said, that which kind of the cells or how many cells you are going, but is there any kind of the, I mean that line that at least we can say that, okay, if we keep it for this threshold, we are sure that at least, we are gathering majority of the cells in the platform, something like that? Or is there any, I don't know, programming or something like that we can write down in our order script to say that, okay, exactly what we are looking.
So that's a good question. So Edmonton's asking, how can we set this threshold? Is there any rule we can use? So there's no absolute rule. There's been many discussions in the single cell community that there should be a mathematical definition for what a cell type is that we could apply to define our clusters. And every time people get together and try to define it, everyone disagrees and no one reaches a conclusion. So we don't have a rigorous definition for what a cell type is. As a rule of thumb, I like to say if you have at least 50 marker genes that are different in that cluster compared to all of the other clusters, that's probably a distinct cell type. So Ottawa is asking how the silhouette index compares to the Dunn index. I'm not actually familiar with what the Dunn index is. Oh, so it's another metric for evaluating clustering algorithms. So it looks very similar. It's slightly different calculation, but it's a very similar idea to the silhouette index.
Would that mean that even this silhouette index score is, it's not a hard rule that it has to be like 0.65, it can be lower as long as we meet the other criteria, the marker genes?
Yeah.
So we can lower it.
Yeah, so you can lower the silhouette index. The silhouette index is pretty good if you have no idea what you should be looking for. But in practice, it doesn't actually work that well for biological data because we have subtypes. So CD8 T cells and CD4 T cells will generally have a pretty low silhouette index because they're quite similar to each other. But biologically, we know those are important cell types to distinguish from each other. So it can be a little bit tricky. Often the silhouette index will underestimate the number of real cell types there are because we have all these subtypes. But it's a good thing to have in your toolbox because it's very common for reviewers to ask, oh, how did you pick your clustering resolution? So that's why you have all of these different ways of measuring it. So you can go through a bunch of them and say, hey, we have this objective score that suggests roughly this number of clusters. When we annotate the cell types and use marker genes, that agrees with that score. Therefore, this is the correct number of clusters. So we have another question from Ottawa. Are the clusters in UMAP necessarily related with silhouette index? Could we find exceptions? Yeah, so the visualization of the clusters in the UMAP aren't necessarily related to the silhouette index, but you tend to see that clusters that are overlapping each other within the UMAP will have lower silhouette indexes than those that are not. So that is most of the time, I'd say like 99% of the time you see that. But when you get into more fine grained than those two of like they're overlapping versus completely not overlapping, it won't necessarily correlate perfectly with the silhouette index. So that's why you want to use the silhouette index and not necessarily the UMAP, because you can also have cases where the UMAP, this will show the points overlapping each other, but you get a good silhouette index. And that happens if essentially you have a dimension of variation in your data set that the UMAP failed to accurately separate from the others. And essentially there's a vertical dimension that's trying to stick out of your UMAP that it can't show, and it's squashed those on top of each other. So it looks like all the points are mixed together, but they're actually distinct. but they're in a dimension you can't see on the UMAP. So we've had that happen a couple of times, but it's fairly uncommon. Another question on the previous slide, does the coefficient value threshold account for the shape of the silhouette? Silhouettes, is the shape of the curve relevant at all to determine the two clusters are distinct enough to separate? So here, the shape of the curves is related to the distribution of the silhouette scores across the cells in the cluster. It's not super relevant in terms of the interpretation of the silhouette index. Usually just the average score for the cluster is just as good, if not better. Occasionally, you can find places where the clustering algorithm has made a mistake by looking at these silhouette profiles, where you can occasionally see sort of a set of cells that has a high silhouette index, and then another set that has a low silhouette index, which usually indicates the clustering algorithm has incorrectly assigned those cells with the low silhouette score. That can happen usually mainly if you have a really small cluster, because the clustering algorithms tend to like all your clusters to be about the same size. So if you have a really, really tiny cluster with like 20 cells in it versus clusters with hundreds and thousands of cells, it's very common for the clustering algorithm to incorrectly assign some extra cells to that really small cell cluster. And then you can see that on your silhouette profiles. Another question from you, Ottawa, is best to start with the highest resolution and then move to lower resolutions, or the other way around? Oh, I ask because sometimes it influences when you do the annotation. I wouldn't say there's a better way of doing it. It's more up to you and what you're most comfortable with. We tend to, sort of the easiest way for annotation purposes is often to start with a higher resolution than you need. And then as you're annotating your clusters, see that you're getting a whole bunch of clusters you're annotating as the same cell type. So you might find you have 20 clusters that all look like they're T cells. And then you can increase or reduce your resolution until those guys merge together. Or you can simply by hand say, oh, we're going to merge all of you guys manually together because you're all the same cell type and I can't tell you apart. So starting at higher resolution tends to be quicker to getting you to your final, most precise cell type annotations than going from a low resolution to a higher resolution.
So if you have this subset of T cells, you want to like subclusters of those T cells, can you then apply silhouette for subclusters, or you have a sentiment?
So for Ottawa, we're at, Edmonton is asking about how you do subclustering and whether you can use silhouette for the subclustering. So yes, you can. When you do subclustering, generally the best approach is to separate that cluster from the rest of your data set and rerun everything. So you can still do the silhouette index, you can still do your highly variable genes, redo your PCA and your clustering. You can redo all of it. So Edmonton's asking, is it possible to have clusters that you can't define what it is? Sometimes. So when that happens, either you've discovered a new cell type, and then you have to sort of try and figure out what that cell type's doing biologically, maybe doing validations or something like that. Or it can happen if you have poor quality cells in your data set and they're forming a cluster. That can also happen. Or it can be doublets. So they look halfway in between two other cell types and you can't tell which is which. So that can also happen. Okay, I think I've got all the questions. Okay, yeah, so that was clustering. So now we'll talk about annotation. So once you've got your clusters, you need to figure out what cell types they represent. And although I'm sort of presenting this as a linear process from clustering to annotation, often you bounce back and forth between these. So do a set of clusters, you'll try to annotate them, find you have some clusters that are either the same cell type or you can't figure out what they are, and then you'll adjust your clustering parameters to either try and break them apart or merge them together, and then annotate them again. So there's many different ways to do annotation. The main overall ideas for it is to either do automatic annotation, where you use some sort of algorithm, and reference data. So you must have a reference for this. In best cases, you have a reference single-cell RNA-seq data set that has cell type labels already. And then you can train an algorithm on that to label the cells, and then apply that algorithm to your data set and have it automatically label the cells for you. You can also do it manually, usually by looking at marker genes for each cell type and using your own biological knowledge or looking them up in the literature to figure out what cell type that is, one by one, manually. So I'm not going to go through automatic annotation tools. There's got to be like 40 of them now out there. The most popular ones are SingleR, because it has a pre-built reference for both mouse and human that covers most cell types. It's not perfect for all organs, but for the most popular tissues like cancers or PBMCs, it has pretty good references for those. But there's lots of other ones available. For manual annotation, last thing on automatic annotation before I move to manual annotation is, while the algorithms are pretty good at general cell types, so they're very good at classifying a T cell versus a macrophage, fine cell types they tend to be less accurate on. So even when you do automatic annotation, often you will then have to do a step of manual annotation as well to confirm the cell types are correct, as well as to check for cell subtypes and label any clusters that it was unable to annotate. Because while they're accurate for T cells versus macrophages, they're much less accurate for Tregs versus CD8 positive T cells. So when you're doing your manual annotation, which is where everyone eventually ends up, you're using marker genes. So just be aware when you're using marker genes that even the literature is not always that reliable. So if you look in the literature, both ITGAM and FCGR3B are listed as markers of neutrophils in many papers. But you can see in ITGAM, it's actually expressed in many myeloid cells, whereas FCGR3B is specific to neutrophils. So when you're using marker genes, even those found from the literature, you need to be a little bit careful. Check whether those marker genes are sufficiently highly expressed that they're reliably captured in your RNA-seq data. So there are some marker genes that are very popular in the literature, like CD4, which aren't captured very well with RNA-seq for whatever reason. Consider how high is it expressed. Is it expressed high enough that you can really classify your cells on it, or do you need to look at sort of the whole cluster level? How specific is it? Is it really only expressed in that cell type, or is it expressed in multiple cell types at different levels? How big is the difference in the log-full change for that gene in the main cell type it's a marker of, and other cell types it might be expressed in? And also consider how many markers you're using. Single-cell RNA-seq data is noisy. Using one gene to annotate a cluster is often not a good approach. You generally want to be using multiple markers and show that they all agree that this is the annotation for that cluster before you conclusively say that yes, this cluster is that cell type. Now, Ottawa is asking, is there a way to direct the clustering? Say if you're comparing two conditions and you expect the cell types will be different. Not really. The ways you can sort of tune your clustering is by altering the highly variable genes you're using. So you could add in disease-specific genes to there if you want to try and push the disease clusters apart from the healthy ones. Or you can fiddle with the K and the resolution. That's really all you can do to control what the clustering algorithm is doing. There's no way to tell it, you expect the clusters to look a certain way. Yeah.
You mentioned like 50 marker genes. Is there a way, instead of like individuals showing certain graphs, to like manually select 50 markers and then say, oh, here's the cluster, and then combine bar graph, I guess. No.
Yeah. So the question is, how do you show lots of markers at the same time, right? Essentially. So there is plots called a dot plot, which shows your clusters on one axis, your genes on another axis, and gives you a dot where the size and the color represent how highly expressed that gene is in that cluster. So you can use that to show up to 20 or 30 markers at a time very easily. You can also use things like a differential expression test between clusters to find marker genes de novo without needing to look in the literature to find them. So you could ask, what are the genes that are most different between these clusters? And then look at what those are and try and figure out what the cell type is based on that. So Otto's asking, can you use negative gene signal to help you annotate your clusters? You can, but it's more risky than using a positive signal. Because for a negative signal, you never know if that gene is truly not expressed or if you just didn't detect that gene. So that gene might be still present, but at a low level, and you just happen to not measure it. So it's a bit more risky. So another question, would you do that for one cluster versus all the rest of the cells, or for neighboring clusters, the differential expression test? Typically you do it for the cluster versus all the rest of the cells, but that's only good for finding general cell types. If you're looking at specific subtypes, it's better to do it that cluster versus the neighboring clusters. Because if you do one cluster versus all the rest of the cells, all your T cell clusters will say, oh, these generic T cell markers are all different, and they'll all look the same. But if you then do each of those clusters versus their neighboring ones, you'll see, oh, these ones have CD8, these ones don't have CD8. All right, so for the last topic for clustering and annotation is what you do if you have multiple samples. So these days, typically you'll have lots of samples, often from multiple conditions. So how are you going to annotate them? So you could take them one by one and cluster them, and then annotate the clusters in each of the samples, and then align those one by one across all of your samples. But that takes a lot of time and a lot of effort. So most often what you'll do instead is you'll merge or integrate the samples together and find a set of clusters that are shared across your samples, and then annotate those clusters. And we're going to talk about integration at the end of the day today, how you do that. Okay, so any more questions about annotation? Yeah, so I was asking, if you're interested in a specific cell type, is the way to isolate that cell type based on no markers, then increase the resolution just for that group to identify subclusters and differential expression between those clusters? Absolutely. Yeah, if you're looking for a specific cell type or a specific subtype, you can just subset out the approximate cluster, and then rerun your entire clustering algorithm on that subset. And there's one more person typing.
And just a heads up, for 12 minutes from scheduled break.
Or 12 minutes from scheduled break. Then I'm going to call off questions, and we'll come back to questions because I want to finish differential expression before we go on break. You can leave your questions in Slack and I'll answer them when we have time. So now we're going to do differential expression. So there's two main types of differential expression you'll do. One is between clusters to annotate them. The other is between conditions. Okay. And the way we do that is different. So for finding marker genes, mostly what we're asking is what genes are unique to one cluster, or what genes are different between two clusters. So when we're doing this, what we care about is not necessarily to find every gene that is differentially expressed between these clusters. We're more interested in finding genes that are specific and the ones that are fairly significant, but mostly ones that have the biggest difference, right? We want the genes that are very different. We don't necessarily care that much about precise significance scores or being super sensitive to find every gene. We just want to find what are the biggest differences. And because we're going to be doing this for a lot of clusters, potentially at different resolutions, we want it to be relatively fast. So calculating accurate p-values for differential expression between clusters is actually really hard. Because if you imagine this is our distribution of expression values in our data set across all of our cells, after we do clustering, we will have separated that distribution based on those expression values. So we will separate those cells that have a high expression of this gene from those that have low expression of this gene because of the clustering algorithm. Right? That's what the clustering algorithm is doing. This means we're much more likely to have genes to be different between our clusters than if we were just taking a random set of cells. What this means is we get inflated p-values. So our p-values look much more significant than they really are because we've biased our test for finding differentially expressed genes, right? So when you pre-filter, you can also pre-filter genes based on the log-fold change to speed up your p-value test. So this is what Surette does by default, and this further biases. So you first check which genes look like they're differentially expressed, and then you test them. Well, now, of course, you're going to get low p-values. You've already biased towards that, getting that. So it's actually quite hard to get statistically accurate p-values. If you do want to get them, the paper here has a method that calculates them accurately and accounts for these biases. However, most of the time you don't really care. Right? You don't care if the p-values are exact. You're just looking at the top 50 most different genes anyway. The p-value of those doesn't really matter. So doing the actual exact p-value test usually isn't necessary. Instead, we do something fast and easy and universally applicable. And that's a non-parametric test called the Wilcox rank sum test. So cross-testing, it works pretty well, similar to many more complicated methods. It has some nice properties in that it's independent of the distribution of your expression values. So if you do integration, if you do batch effect correction, if you do normalization, doesn't matter which steps you've done, this test will work. It doesn't make any assumption about what your data looks like. So it can be applied to batch corrected, scaled, normalized, regressed, whatever data. Any normalization you want to use, you can use it and this test will work. However, it doesn't account for confounding factors. So you do have to normalize the data first before you use this test. That's all I'm going to say about that because it's really simple. We're generally not doing anything fancy there. But what we're usually most interested in is actually our case control D, so comparing between different conditions. So for instance, we might want to ask, what's the effect of a gene knockout on microglia? So in this case, we'll typically do an experiment where we have multiple replicates, so multiple mice with each genotype or in each condition, or maybe this is patient data. And then we'll do single-cell RNA-seq on each of those mice and get a whole bunch of cells from each mouse. And then we want to compare these groups to each other. So if we're comparing our gene knockout to our wild type, what should we be using for the sample size? Should we be using the number of cells we have or the number of mice we have? Yeah, so we should be using the different mice. So we shouldn't be treating each cell as a biological replicate. We should be treating each mouse as a biological replicate. So in this case, we would have a sample size of 3 in each group. So the way we can do this most simply is what's known as pseudobulk. So the pseudobulk idea is we've got our single cell data, but we're going to turn it into bulk RNA-seq data. And then we're just going to use standard bulk RNA-seq differential expression methods on it. But because we've done it for single cells, we can do this for each cell type separately. So we can essentially in silico sort our different cell populations, turn it into bulk RNA-seq, and do bulk RNA-seq differential expression for each cell type for our different conditions. So we aggregate the expression of all of the cells of a particular cell type in each biological replicate. And then we use dSeq2 or EdgeR or your favorite bulk RNA-seq differential expression method to test for differences between our group. So these ones are based on a general linear model, so both dSeq2 and EdgeR are. So I'll explain what that means and how you can use it to ask your specific question. So it's a general linear model. So we're going to forget about that complex name. We're going to talk about linear regression first, because hopefully everyone knows what linear regression is, right? We have something on the X, something on the Y, and we fit a line between those two. So we can rewrite this as, instead of our y equals mx plus b, y equals b0 plus b1 times x, where our b's are now called coefficients. Right? That's just some terminology we're going to use. So in this case, our x and y are continuous values, but what if our x is discrete? So let's say our x is instead whether something is pathogenic or is not pathogenic. So we have our mutation or we don't have our mutation. In this case, we still want to be able to use our linear regression, so we just change our no and yes into 0 and 1. Now our x-axis is numbers, and we can do linear regression again. Okay, so we're still just doing linear regression, but now we've got discrete groups. So now when we do our y equals mx plus b, our b0 is the expression in our 0 group, so our reference group. B1 is the difference between our pathogenic group and our reference group. So what if we have more than one thing we want to test? So we could have a experimental design where we have different combinations of 2 gene knockouts. So we have APP knockout on one axis, ApoE knockout on the other axis, have our reference normal wild type allele as 0 and our knockout as 1. So you can see we have 4 groups of cells now for the expression of any particular gene. So now we can just add an extra term to our linear regression. So now we have B0 plus B1 times X and B2 times Z, where X is APP knockout and Z is ApoE knockout. And we can just fit this model to our data. And now we're doing two-dimensional linear regression. So our B1 is the slope of those two lines, right? That are the difference between APP wild type and APP knockout. And B2 is the slope of these lines. That's the difference between ApoE knockout and ApoE wild type. And our B0 is the group that's wild type for both of them. Okay. We could also have a case where we have an interaction. So maybe our group that's APP, both a knockout for APP and ApoE, is not just the sum of APP knockout and ApoE knockout. So we can add an extra term, B3, to account for that. So we just add B3 times X times Z. So now if X is 0, we don't use B3. If Y is 0, we don't use B3. We only use B3 if X is 1 and Z is 1. So we have both knockouts at the same time. So B3 is called the interaction between ApoE and APP. Right, so if we're doing our pseudobulk expression for this case, we'll convert our data into a gene by mouse matrix for each cell type individually, and we'll apply our model and fit it and test each of our B1 and B2 to see if they're significantly different from 0, to see if this gene is differentially expressed with APP knockout or ApoE knockout. I think we're going to go for lunch because we're experiencing some noise disruption here, so we'll pick up after lunch. All right, so we just finished option we can use for differential expression between groups in single-cell RNA-seq. And that's using what's known as a mixed effect model. So this has a lot of complicated math behind it. I'm going to try to explain it without any math whatsoever. So the idea here is we've got our two conditions, control and disease, and we've got a bunch of cells from multiple different replicates of each of those conditions. And we believe that cells within a replicate are going to be similar to each other purely because they belong to the same replicate, right? They came from the same mouse or they came from the same human being. So they're going to be more similar to each other than cells between two different mice or between two different humans. And we want to account for that in our model so that we're not overestimating how much power we have in our test, right? So we're not treating every cell as a completely independent biological replicate. We need to acknowledge the fact that we have these 3 replicates and multiple cells within each replicate. So the way we do that is with what's known as a random effect. And we tell the random effect in our model what each of our batches are. So we tell it all of these cells in orange belong to replicate orange. All these cells in yellow belong to replicate yellow. All the blue belong to replicate blue. And we say, we want to allow you to have a random effect in the model that accounts for the differences between our different replicates. So the difference between our blue cells and our yellow cells to the orange cells will be modeled by our random effect. And this random effect, we're not going to get an actual parameter for this, so we're not going to get a coefficient that tells us what that difference is, but it will be incorporated into our model, so that our model is treating these as 3 replicates with multiple cells in each replicate. We then have what's known as fixed effects, which are regular coefficients that we were talking about before. So our difference between our control and disease are what's known as fixed effects. So they have a coefficient associated with them that we're going to estimate. Okay, and we have to do it this way because our replicates are confounded with our fixed effect. So if we tried to estimate a coefficient for each of our replicates, so our yellow and our orange and our blue, all of our fixed effect would disappear because that fixed effect is also present between our different replicates. But by using this random effect that doesn't estimate that parameter explicitly, we can model that variation between replicates without sacrificing our fixed effect. So we can keep estimating our fixed effect correctly. Okay. So a mixed effect model separates the variability between batches and the variability between conditions, and it accounts for the non-independence of cells within each batch to allow us to test for a difference in our fixed effects accurately, even when we have multiple cells from multiple replicates. It does assume the difference between replicates in your control group is roughly the same as the distribution of differences between replicates in your disease group. So it's just an assumption of the model. Generally, that's going to be true for your data sets. There might be rare occasions where that's not true, and then you'll not want to use this type of model. So this can be run using Nebula or MAST. I don't think we're going to actually cover this in the lab, but these are pretty simple to run. Especially Nebula is very easy to run. It's one command, essentially. Okay. So in terms of our mixed effect differential expression model, we take our cells from our multiple replicates, we keep our cells by gene matrix, but we label each cell with what replicate or what mouse it came from. And then we put it into this model where we have our fixed effects. So we have our B0, our B1, and B2. And then we add a random effect for our batches. So we give the model the batch ID for each cell. And again, we're simply testing whether B1 and B0 are significantly different from 0 to see whether our different conditions are having an effect on our gene expression. So I have another complex example, which you can work through on your own time, because I want to move on to the lab to make sure we have time for the lab. This is just an example where I have a little example of how you would do the analysis using pseudobulks, or how you would do it with a general linear mixed model. So that's what GLMM is. So just in summary, we talked about how single-cell RNA-seq uses graph-based clustering because of its scalability and good appropriateness for high-dimensional data. Talked about picking an appropriate clustering resolution and how hard that can be, and how we'll often go back and forth between clustering and annotation. We talked about automatic annotation being fast and good for general cell types, but the necessity of manual annotation when you're looking at specific subtypes or rare or novel cell types that might not be in your reference that you're using for your automatic annotation. And then the importance of accurately representing your biological replicates, whether using pseudobulks or mixed effect models. And that we can use general linear models to estimate effects of multiple different causes simultaneously. Okay, any questions